Chapter 28 is pregnancy and childbirth. This is a really big topic, so we are going to spend a fair bit of time talking about it. A lot of different things go on, and many of them are beyond my understanding, but what I do know, which is more than I would like to probably, I'm going to share with you. What we see here are we see the general outline of what's going to happen in this process of growing a baby. So we start out with the idea of fertilization, which is a single sperm getting into and fertilizing a single egg. Then it grows and at some point is a ball of cells. Then at 3 weeks or so, it starts to be called an embryo. At that point, it is starting to get some pieces and parts assembled, starting to get some degree of recognizability going on there. And then at 5 weeks, as you can see, 5 weeks is a giant head, small body embryo. 8 weeks is still a large head, but the body is growing a little bit more proportionate to it. And then up to 12 weeks. So in 12 weeks, an incredible amount of stuff goes on. We go from one cell to many millions of cells. We go from an unrecognizable ball of stuff to a very clearly baby human, no question about that, and all of the pieces and parts are present in that 12-week-old baby. Not all of them necessarily functioning yet, but at 12 weeks, all the pieces and parts are laid out. So an incredible amount happens in that time frame. So this is really the first 3 months, so the first trimester of the pregnancy process. And because this is where so much stuff is going on, this is where the primary opportunity for mistakes occurs. So if something is going to go wrong with the pregnancy, it will likely go wrong in that first trimester. And I remember very clearly when my wife was pregnant with both of our children, we made the conscious, deliberate decision to not tell anybody that my wife was pregnant until after the first trimester was completed. And our thought process was that because most of the things that go wrong in a pregnancy go wrong here, let's not tell people until that highest risk stage has been completed. And after that, if everything's still going fine, then we'll go ahead and tell people. And our thought was we don't want to have to tell people we're pregnant, and then, oh, wait a minute, no, something went wrong, miscarriage occurred, and scratch that. That was our personal decision. Each individual obviously has the opportunity to decide for themselves how they want to approach that notification thing. I've heard people do it within days after they find out they're pregnant, all the way up to they don't tell until they're asked and can't hide it any longer. That's a personal thing that would be up to the individual. But again, this is the highest risk stage of pregnancy because this is where most of the big stage concepts are taking place. Let's briefly go through the steps of fertilization. What we see here is we see the egg, and the egg is surrounded by those layers of cells there. And if you have watched the video for childbirth and pregnancy already, they would talk about the zona, and these layers of cells are the chaperone cells they would talk about, and then underneath that is the hard shell called the zona. So these cells basically are there to protect the egg, isolate the egg from its environment after it's been ovulated, and really it provides the sperm another hurdle that they have to cross to fertilize the egg. In all the millions of sperm that were produced, ejaculated, and are swimming perhaps towards the egg, you really want the best quality sperm that's possible, which is probably going to have the best genetic components inside it that the father had to offer. So if you put this layer of chaperone cells there, that forces the sperm to dig and chew their way through, ensuring that weak sperm that don't have much energy left at this point don't get through to fertilize. So it's another way to be discriminatory about what sperm is allowed in. Once that sperm gets inside, so that one sperm that will fertilize the egg, we have here the male pronucleus and the female pronucleus. And down here at the bottom, we have 3 different polar bodies. So remember in this process that the egg went through a couple of stages of meiosis here, and it's just now, once it's fertilized, finishing meiosis 2. And in that process, the polar bodies were basically the other 3 copies of each of the chromosome that you would have had early in the meiotic process, ensuring that the egg itself in the female pronucleus only contains 1 of each chromosome. The sperm contributes 1 of each chromosome, and that gets our egg back up to, at this case, not necessarily an egg anymore, it's a zygote because it's been fertilized. And at that point, we've restored the diploid condition. So that would be 2 of each chromosome, so a pair of each of the 23 chromosomes. So the sperm by itself had 23 chromosomes, the egg by itself 23. Now it has 46, the 23 pairs that the original cells that made the egg and made the nucleus had to begin with. So we've undone a lot of things just to redo them to make sure the next generation doesn't have too much. The next step here is that the genetic material from the sperm and from the egg are getting closer to each other. They're about to join, and basically here at this step, once the sperm and egg genetic material has combined, then we're officially at the zygote stage, and so now we're a distinct entity that has never existed in that genetic format before. In a real cell egg-sperm combo, it would look like that. So the polar bodies down at the bottom, and then the male and female pronuclei fusing in the middle. There are a couple of things in place in this arrangement to make sure that only 1 sperm fertilizes the egg. You don't want polyspermy, so you don't want more than 1 sperm entering the egg. If you do, there are going to be serious problems that come from that. So at this stage of development, let's say 2 sperm did contribute their genetic material. That would then give you 3 of each chromosome present inside the egg, instead of the 2 you're supposed to have when egg plus sperm finally gets back up to 2 of each. 3 of each would be called trisomy, and if you had 3 of each one, that is immediately going to result in death of the cell. So it can't handle 3 of all the different chromosomes present. There's room for it, but it just isn't compatible, and at that point, nothing works. Now, sometimes you have a single chromosome that exists in trisomy condition inside the cell. So let's say either the sperm or the egg made a mistake along the way and kept an extra 1 of the chromosomes it was supposed to have, and by random chance didn't die or have other issues before that happened, then you might have 3 of one particular chromosome, and sometimes that can survive. It's never going to produce an outcome that is the best possible outcome, but it may be survivable. And probably one of the more well-known of those is called trisomy 21, in which 2 of chromosome 21 are present, typically in the egg, although it could be the sperm. But it's usually the egg that this occurs with. So the egg failed to eject its second chromosome 21 when it went through ejection of the final polar body. And then the sperm added its 1. So the 2 probably from the egg, 1 from sperm would give you 3 of that particular chromosome. And if you only have the extra of that one and not of others, that's something that the egg and eventual zygote can probably survive. But it will result in what is generically termed on the street Down syndrome. And we could talk about Down syndrome, and we'll do that in a later chapter when we talk about genetics. But it certainly is a condition that, while there are much worse conditions, if it's avoidable, certainly one would want to do that. And so just while we're here, the quick and dirty explanation for why trisomy 21 occurs and who it occurs most often in is older mothers. So probably past the age of 35 is where you see the highest frequency of babies born with Down syndrome. And that's simply because the egg is now let's say a 37-year-old mother. That egg is now 37 years old. And so you're expecting a 37-year-old egg to come out of its hibernative state, resume full function, with no mistakes or hiccups along the way. That's like parking a car in the garage and coming back 37 years later and expecting it to start on the first try. Could it do it? Sure, it's always a possibility, but the older it is, the longer it's been sitting around, the less likely it is to work well. So my personal recommendation would be that childbearing occurs between the ages of 25 and 35. I would propose that biologically that's the best timing. After the age of 25, mama's body has built itself and it's ready to make another one. After the age of 35, things like Down syndrome and other genetic abnormalities start occurring much more often, and that's directly because of the older egg that you're working with. I said these could come from the sperm, so the sperm are not beyond the possibility of making a mistake, but it's not as likely because the sperm are never more than 3 months old. If they've been sitting around for more than about 3 months, they're going to be disposed of and be replaced by new sperm. So they don't have the opportunity to get old and lose function nearly as much. So anyway, we're back to this idea here of we only want one sperm in there. So one of the blocks is you have to chew through those outer layers of cells. And they're labeling it here as corona radiata, or the chaperone cells. And if you chew through all those, the weak sperm fall out along the way. You've cut down the number. Probably 50 or more sperm are going to contribute towards getting through the chaperone cells and then getting past the zona, so dissolving their way past. And to do that, as you would have seen in the video for pregnancy and childbirth, they release enzymes that digest through, but it takes multiple sperm to achieve that. So whoever happens to be the lucky sperm that gets through the zona and through the chaperone cells first, then has another hurdle, and that is the oocyte plasma membrane. And you saw in the video that the sperm had to match up with the proteins on the egg surface for the fertilization event to occur. So here's these little blue structures right here. Those are what have to match up with the cap of the sperm. And once that happens, now the sperm has permission to attempt to fertilize. So here we have the sperm chewing through the zona, lining up and binding with those proteins, and then injecting its nucleus, basically launching its nucleus inside the egg. Now the video sort of made it look like the tail of the sperm went with the nucleus of the sperm. That's not really the case. Really just the head and a little bit of the collar will go in, and most of the tail probably is going to remain outside. But what happens is immediately some reactions occur. And there's a slow block to polyspermy and a fast block to polyspermy. And what we might call the fast block to polyspermy results in what you see at step 4 at fusion. You see the membrane starts to be distorted. So once that protein lines up between the sperm and the egg, it distorts the surface with those proteins, making it very, very difficult for other sperm to line up properly. Then as soon as the sperm injects its nucleus into the egg, all of those proteins, as you see in step 5, fall off. When those proteins are no longer there, well, nothing to identify with, nothing to attach to, so the other sperm don't recognize that they're close by. Then the zona where the sperm had chewed its way through, the egg shell here, goes ahead and rehardens and becomes, in a way, what the eggshell of a chicken would, impermeable to most anything else that comes along. So the fast block is distort it, then start removing the proteins. The slow block is finish removing the proteins, reharden the zona, and remove the identifiers from the zona, so the sperm, when they come along, even if they come along in huge numbers, don't recognize the egg as being what they're looking for because all the name tags have been removed. So those things, both of them happen fairly quickly, and again, the point is to make sure that only one sperm comes in. If we briefly follow the process this egg will go through in these, we have ovulation from the ovary, Then up at what I previously labeled as the wrist of the fallopian tube up here at this first curve, if the egg entered the fallopian tube here, ideally the sperm meet it somewhere around that wrist. Let's assume fertilization occurs, and within 2 days, we have gone farther down the fallopian tube here and divided twice. So we went from 1 cell to 2 cells, then to 4 cells, and you saw this very nicely in the video. And as you go down the fallopian tube, you have more and more divisions until by the time you get down here and enter the fallopian tube, you are a ball of cells, containing perhaps a couple of hundred cells. At that point, you have to get out of the zona. So when they were talking about the ball of cells orchestrating its escape from the zona and entering the uterus, you're doing that. So the zona was there protecting, which that was fine, but you can't implant in the uterus if that shell is intact. So it has to get out of the shell. At that point, it's vulnerable to the mother's immune system. Mama's immune system could see a egg with the zona intact all day long and ignore it because it says, I recognize zona pellucida. That's been here from before I was born. That's an egg, and it doesn't recognize that inside the egg is dramatically changed. It just says the outside looks like an egg's supposed to look. No big deal. Don't pay attention. And really, there's nothing for it to attach to or attack on the shell. But as soon as it gets out of that shell, it's now a ball of cells that originated from a sperm-egg combination. And that particular combination has never been seen before by this mama's immune system, and the immune system would then identify it as a foreign particle and try to kill it. So it's a very dangerous time for this ball of cells here, going from the zona shell and implanting in the uterus. Once implantation completes into the walls of the uterus is now enclosing and protecting that ball of cells, and the immune system doesn't see it at that point. So this is where probably a fair number of fertilized eggs die in the process, is the transition from protective shell to implantation, and mama's immune system finds it and kills it first. Implantation needs to occur because there need to be nutrients. Up to this point, the egg has used about all the nutrients that were there, making all these new little cells. And so the uterus is designed to be a stretchy environment that can replenish nutrients. And so one of the first things that are going to need to happen after implantation is tap into that new nutrient source. So if we look at implantation up close here, in picture A, it comes in contact with the lining of the uterus and sticks. Then it starts burrowing its way in and sort of making a hole for itself, and at some point it gets sucked all the way in. The video had a fabulous image or action there of the egg sucking into the uterus and disappearing, and at that point it has successfully avoided mama's immune system. A lot of things are going to happen to mama during pregnancy, and one of the most significant are the changes in hormone levels. So we talked about hormones when we talked about ovulation and we talked about the menstrual cycle, but if we go through the entire process of pregnancy here, one of the things we see is that if ovulation and fertilization happened again around day 13 or somewhere in that ballpark, so around week 2, let's say, in the pregnancy. And this is an interesting thing, is that you're pregnant and the first 2 weeks of the pregnancy don't actually really count as the weeks of the pregnancy. They do and they don't, but you weren't actually pregnant for the first 2 weeks, even though as part of the 40 weeks, that's part of the math equation there. So what happens at and shortly after ovulation and fertilization is human chorionic gonadotropin increases dramatically. Estrogen and progesterone are going to increase throughout the rest of the pregnancy out to the idea of birth, but relatively slowly through most of that. But HCG spikes very dramatically in the first couple of weeks, and this is what's going to be involved in the pregnancy test. And I think we've talked about that just a little bit before. But the pregnancy test is looking for hormone changes throughout the body, and a big spike in HCG is typically only because of a pregnancy that's happened, and therefore that's what the test, when you do an at-home pregnancy test, and at the doctor's office for that matter, is doing is measuring HCG levels to see if they've gone past a certain point. And there aren't very many other explanations for HCG above a certain point in that scenario. Those levels will then go down to a relatively stable level for the rest of the pregnancy. Estrogens and progesterones, again, for the most part, increasing steadily throughout the process. As you can see here, progesterone will drop off a little bit towards the end, but for the most part, it was climbing the whole way. Anyone who is a female or has been around a female for more than a few minutes will understand the idea that changes in hormone levels change how really anybody feels and behaves. And in a female, there are much more dramatic hormone level shifts back and forth. And during pregnancy here, you can see that HCG went crazy. Then estrogen and progesterone going up. And so all of these things becoming somewhat unbalanced in an appropriate way, but still that will cause a person's feelings throughout a pregnancy to change. And so at some stages of the pregnancy, perhaps they're the tired, exhausted, don't feel like doing anything stages. And then at other stages, you get to the what would sometimes be called the glowing, happy, energetic, enthusiastic stages of pregnancy. So these differing levels of hormones throughout this experience are going to change how a person feels and behaves. And it's to be expected. It might not necessarily be fun, but it's perfectly normal. If we look here at this process of getting nutrients, we said that this zygote now has used most of its available nutrients, and so it needs an additional nutrient supply. So that's what the placenta is. So the placenta really is a nutrient source that comes from mama's blood, and as long as mama's blood is there, it is an unlimited nutrient source. So early in the burrowing in of the zygote, or the implantation, there are blood vessels in the endometrium, and signals coming from that burrowing zygote are going to stimulate those vessels to grow towards it. And so what you see here by probably step B and certainly step C here, the 12 and 16-day stage, blood vessels start growing towards it, and the ball of cells becoming the baby starts to differentiate as well. And at day 16, it's called an embryo. At this point, it's getting most of its nutrients from mama, and what you see are chorionic villus, or chorionic villi, which the video spent a great deal of time on. And what it does is it basically allows mama's blood to come up and touch it, but mama's blood should never cross into the chorionic villus and then enter the embryo or eventual fetus. So this is an option for baby to pick up nutrients and oxygen from mama, drop off waste products, carbon dioxide, that sort of thing, back into mama's blood, because the baby has no other source for those things. And also keeping mama out. So mama doesn't technically, from an immune system perspective, realize that this technical parasite is present, because as long as the placenta keeps mama's blood and baby's blood separate, everything's fine. A couple of days after that, you get to the 4.5-week embryo at the bottom left here, and at this point you see we've got arms and legs growing, we've got head and tail discerned, and by the way, babies early in the process have tails and have gills, just like a fish or any other vertebrate would have. So at those stages, at the, say, the 4.5-week-old embryo, a baby human, a baby chicken, a baby rat, and to a certain degree, even a baby fish or lizard wouldn't look much different, because all of them have tails present, they have little limb buds present, they have gills, and so they're all working much the same way at that point. From there on out, though, significant differences do start occurring physically. At this point here, you can see the yolk sac. So that's the remnants of the nutrients that were available in the original egg. So mostly used up, starts going away. At this point, there is a significant blood supply from mama. And you can see here baby being whatever size baby happens to be here, and then all of this stuff around is placenta. So baby is much smaller than placenta early in the process. So that means the nutrient source vastly outweighs, outnumbers, outprovides that growing embryo, and that's okay. And at this point, a definite umbilical cord is present. So that's the connection between the placenta and the baby. At the 13-week fetus stage here, the placenta has not gotten any smaller. It's actually probably gotten a little bit bigger as you go, but the baby is growing so much more rapidly at this point that the baby starts to become bigger than the placenta. And that's okay, but it just starts to be a noticeable shift in the size of the baby relative to the size of the placenta. Obviously, as the baby grows, the placenta has a harder and harder time providing nutrients, and so perhaps one of the final stimuli towards starting the process of contractions and the labor and birth process might be the baby just can't get enough nutrients from the placenta. So you can only provide so much through the placenta, and quite frankly, mama's body can only provide so much to the placenta, regardless of how well the placenta transfers it on. So this is a pretty good system. It's a renewable energy source system, but again, it's really only designed to be in place for a couple of months. If we look at that placenta up a little closer here, over on the left here we have the umbilical cord coming in, so that's plugging into the baby's belly button area, and these arteries and veins come over here, and here are the projections coming from the placenta from the baby's perspective. So lots of blood vessels out here with a very thin layer of tissue around them. They're picking up nutrients from mama's blood dumping in here, so they're picking up oxygen and nutrients by diffusion into this circulation and then back into the baby for use. The waste product gets dumped out into the mama's blood circulating around out here in the open areas of the placenta and gets picked back up by mama's veins, taken back through mama's system and cleaned just like it would mama's own blood normally. So this is a very intricate and complicated setup here, but it works. You know that you have skin and you have hair and you have bones and muscles and things like that. And again, the video did a nice job of this at looking at where do those tissues originate from. So this is just a little bit of a reminder. You start out with this ball of cells and it grows a mass down the middle. And you have the blue layer of cells that's there all along. It starts to fold underneath itself, producing a second layer. In this case, the yellow hypoblast. After that's formed, now you have two layers of tissue instead of just one. Then the blue, the ectoderm, it's now called at this point, and endoderm is what you would call the yellow tissue at this point of 16 days and beyond. The ectoderm keeps folding, and it forms a third layer of tissue. So in a way, this is like the human body. You have an outer coating, you have an inner lining, and then you have stuff in between. So those three layers of tissue are what the embryo really starts out with. Before it can make arms and legs or anything else, you have to have a source for skin. You have to have a source for muscle and a source for bones and a source for blood and all that sort of thing. So these three layers further specialize and become all the different tissues in the body. So really everything in the body came from one tissue, that original epiblast, which differentiated into three tissues, which then differentiated into everything you have. So a lot of steps along the way there, and at this point in the process looking nothing like a human at all. That comes only a few days later. Do notice one thing that happened almost first. Nearly as soon as we had those layers attached or assembled, look at what happened over here at this bi-layered embryonic disc, superior view. We've already decided here's the head end and here's the tail end. So really the first decision about what goes where is determined by picking what's the head and what's the tail. And that happens pretty much the same way for all organisms. Once you've made that decision, then you start figuring out where to put everything else relative to that. But if you don't get the head decision made, you sort of have a problem for the rest of the time. So here's that process again up close. One layer or one type of tissue becoming 2, becoming then 3. If we look at this from the side view here, we see that there is a primitive gut, basically just the connection of this developing cell mass to the yolk sac for nutrients. But it's becoming sort of at this point we've decided, down here to the right is going to be the head end. Down here on the left is going to be the tail. So the first part of this foregut connection to the yolk sac will become the mouth. The end of this hindgut here will become the anus, and everything in the middle will eventually develop into intestines and digestive system. But also what we have here, and really the first system that gets serious about developing, is the nervous system. So we see here a neural tube and a notochord. A notochord is a primitive spinal cord. The neural tube is going to be a wrapping material around that. So that's really the first system that starts developing. Everything else is not really there yet. The digestive system thinking about it, but not really making serious progress. But the nervous system is in high gear at this point. So we've already decided head and tail, and now we're going to start to have brain develop. So the first recognizable organ that starts to develop is the brain. The first fully functioning organ is the heart. So there's a little difference there. The first one that works is the heart. The first one that starts to be built is the brain. And this is really, really important, because this brain development is happening in that first couple of weeks after fertilization, at which point you may not know as the mama that you're actually pregnant. And before you knew you were pregnant, perhaps you would have had no qualms about consuming alcohol. And obviously alcohol consumption is a personal choice, but if you didn't know that alcohol consumption was of a special concern to you, perhaps in complete innocence you would go have a few beers or a glass of wine or something like that. And for a normal person who's not pregnant, that's not the end of the world. But if you happen to be pregnant, and right here in this stage where the brain and spinal cord are really getting sorted out and developed, what can happen is interference with development of the nervous system. So brain and spinal cord might actually suffer developmental impairment because of consumption of alcohol very early in the pregnancy, perhaps before you even knew you were pregnant. So this would be before the first menstrual cycle has been missed, and at which point you might start thinking that maybe you're pregnant. And this unfortunately was the most delicate and sensitive part of the baby's development, was here in this early stage. So my recommendation would be that if you don't want a baby to be born with fetal alcohol syndrome, and trust me, you don't, if you have any questions about why fetal alcohol syndrome is such a problem, just go Google it and look at a few of those pictures that you'll see, and that should answer that for you. But you don't want to do that, so my suggestion would then be, if you might be pregnant, if you might be getting pregnant, if there's any possibility of pregnancy existing or occurring anytime relative to now, don't consume alcohol now. Then you don't have to worry about what may or may not happen before you know that you shouldn't drink anymore. I think most everybody would know that once you're pregnant, you probably shouldn't consume alcohol, because it's certainly not good for the baby. Folks in France often don't necessarily feel the need to avoid alcohol, but then again, there are problems along with that too. So my suggestion is anywhere before, during, after a pregnancy in which it's a possibility, no alcohol, and then you have no worries about that. You can just focus on worrying about the 10,000 other things that could go wrong instead. At 5 weeks here, we've got a general body plan. We've got a head starting to form here. So we've already got brain and spinal cord roughly in place. Now we're going to focus on digestive system. And so you can see the stomach and the liver and the pancreas and all those digestive things coming together there. And you also start to see the respiratory system. And the reason why you see this here is that it is connected to, it's originating as part of the digestive system, and then eventually develops a flap that we talked about with the digestive and respiratory systems called the epiglottis that separates the two systems. But because they originate from the same tissues, and because they actually share common tubing for part of the trip, they're built together here. And it's sort of a convoluted path right now because you go past the stomach and all that, and then you go out to the umbilical cord and the yolk sac. Then you come back in from the umbilical cord and pick up with the rest of the digestive system. Later in that process, when the umbilical cord is separated from the baby, that connective tubing to the yolk sac will be disconnected, and it will go ahead and just seal off, and some of those tissues will become other things. And at that point, the system won't be as convoluted because it will be one continuous tubing rather than deviating out from the body and then coming back in later through the umbilical cord. So the system does get a little simpler after birth here. If we look at this from those 3 layers of tissues perspective, I'm not going to spend a lot of time on this slide, but do notice at the top you just had balls of cells. Then you get in step B the neural groove and the neural fold, the neural crest, so that's the spinal cord forming. Then only 2 days after that, so going from day 20 to day 22, that's closed, it's encased, and now we're starting to focus efforts on other things, so forming the rest of the body. And by the end of week 4, so only about a week later in this process, we've gone from spinal cord, brain, and some other tissues around it to now having formed a couple of other cavities and tubes through the system. So the neural tube with the brain, spinal cords there, the digestive system, it has its own tubing through the middle now, and everything's enclosed in one body. At that point, down at the end here, we also see limb buds, so arms and legs start coming in here. Once we've established the internal organization, then the external stuff comes along. We could spend an entire semester. In fact, in graduate school, I did spend an entire semester in a class called developmental biology. And what we did was we took this process of make sperm and egg, fertilize, make a baby, get it born, and spent the entire semester on that topic. And it was a fascinating topic, but for now, we don't have time to spend that much. Thinking about those layers here, you can read, so I'm not going to read it to you. The blue tissues became certain things, the red became certain things, and the yellow became certain things. So they all carried out very significant functions. Some of them a little bit more exciting, perhaps, and seeming to be more involved or more important, but all of them were very critical in this process. That's part of what makes pregnancy and growing and making a baby such an amazing concept, is that there are so many things that happen, and they happen very quickly for the most part. So it's amazing that any of those things, as complicated as they are, ever work correctly. Then when you consider how many things there are that work most of the time perfectly correctly with no problems at all, it's just amazing that that stuff can work when it's so complicated and it happens so fast. But that's just how it goes, and fortunately, again, it usually goes pretty well. What we're looking at here is a huge difference in the circulatory system of a fetus. So that was all the pieces and parts are there, processed, but still getting nutrients from the placenta. And then a newborn has been disconnected from the placenta. And these changes are not going to happen immediately. So labeling this second picture on the right side here as newborn is a tad misleading. Over the course of a couple of months, these changes are going to occur. But immediately, all of the tubing, so we had arteries and veins coming from the belly button here out the umbilical cord to the placenta, all that's gone. Then we had this tubing that was going from basically the inferior vena cava here below the heart, which we'll worry about later. It was going from there out to the placenta and back, but there wasn't a great deal of circulation out here in the vena cava itself. Deeper than that, there wasn't a great deal of circulation going through the aorta here, so the heart of the baby was basically pumping blood back and forth between the heart and the placenta. But at this point of disconnection, there's no longer a connection out there, so the tubing that was here going between the belly button and the inferior vena cava is going to repurpose. Over here we see it became a ligament. So all these blue boxes here are things that served one function before the baby was born and become something different afterwards. And for the most part, those turn into ligaments. So they were blood vessels. They repurpose as simply ligaments to hold things together after that, because they didn't need to have blood flowing majorly in those directions anymore. So there were some changes up here in the superior vena cava. There were some changes down here actually within the heart itself. So what we had in the heart itself was a great deal of blood bypassing the lungs and going straight back out into circulation again. And that was relevant because the baby wasn't breathing yet, and to send all of the blood out to the lungs and back to the heart would serve no purpose at all. So a lot of that blood was bypassing the lungs, going right back into circulation again. After birth, breathing is now important because you don't get oxygen from mama. So that opening that was there in the heart allowing bypass into the other chamber and back out closes up. This is going to be one of the big changes that you notice more obviously in a baby. When a baby is first born, for the first couple of days, how much does it stay awake? And most babies for the first couple of days probably are only going to wake up for maybe an hour or two, maybe 3 hours of the day. They eat and then they go back to sleep. So wake up, eat, go back to sleep. Wake up, eat, go back to sleep. And that happens over and over throughout the day. And as the baby gets older, what you notice is it stays awake for longer periods of time. And after a couple of weeks, you might expect the baby to stay awake for maybe an hour or 2 or 3 at a time. And the reason for that is improved blood flow to the lungs. I said these changes didn't happen immediately. They changed over the course of weeks and months. So that opening between the left and right sides of the heart slowly seals over the course of a couple of weeks. So for the first couple of days, some of the baby's blood is not going to the lungs. It's not getting oxygenated, and that baby temporarily experiences oxygen deficiency. So the baby can't stay awake because it doesn't have enough oxygen to stay awake. It wakes up, it eats, it passes out. It wakes up, it eats, it passes out. And so as that opening in between the two chambers of the heart closes, more blood goes to the lungs. It's now better oxygenated and actually has the ability to stay awake. It has enough oxygen to stay awake and function for longer and longer periods of time. And once that opening completely closes off between the chambers, full blood flow goes to the lungs, and the baby then starts being able to stay awake for perhaps a couple of hours at a time before having to take a nap again. The rest of the things you see here, the umbilical vein that was going out to the umbilical cord becomes over here some connective tissues. The arteries become connective tissues here. And so all that stuff gets repurposed into non-blood carrying functionalities. So that's really quite a bit of rearrangement and change of function going on within the baby at that point.